Hello, this is Matthew Chia's DNA replication stop motion, and here we have adenine, guanine, thymine, and cytosine, and their respective colors. And it's important to note that this process takes place in the nucleus. Okay, so the first enzyme of DNA replication is helicase, which unzips the DNA by breaking down the hydrogen bonds in the middle. Once the DNA is unzipped, the next enzyme, primase, adds mRNA primers to the DNA sequence. It is where the mRNA primers are that polymerase 3, the next enzyme of the sequence, builds. So it starts from the mRNA primer, and for the 3 to 5 strand, it builds in a straight line because polymerase 3 moves from 5 to 3. As you can see, it builds it. But what I didn't show was that polymerase 2, as polymerase 3 builds, it double checks to see any errors. Here, polymerase 1 is converting the mRNA sequence to a DNA sequence. So that was for the 3 to 5 strand, which we call the leading strand because it goes straight forward. However, for the 5 to 3 strand, we can't have that same liberty. We can't go straight forward. So we must use multiple primers to build this DNA strand. So primase adds multiple primers, and here comes back polymerase 3. And because it builds from a 5 to 3 strand, and this is directed in the 5 to 3 strand, it must move in the opposite direction of helicase. So here you can see it moves in the opposite direction. But unlike the 3 to 5 strand where it just moves straight forward, this polymerase 3 must circle back all the way to the other primer, and then it must build in the other opposite direction of which helicase moved. So you can see that right here. And what you get for this 5 to 3 strand, called the lagging strand because it takes longer to build, are these Okazaki fragments. They're not connected, but they should be. So what connects them is this Jesus penguin. I'm just kidding. No. What connects them is ligase. Ligase connects them together to create one strand. So now we have successfully duplicated DNA. And here, this freeze frame should show you that the DNA that we replicated is semi-conservative in the sense that 1, there's an original strand, and 2, there's a new strand. Hello again, this is Matthew Chia, and these are the nitrogenous bases we are going to be using for protein synthesis. We will replace thymine with uracil for mRNA. This process takes place in the nucleus like DNA synthesis. The first step of protein synthesis is transcription, and the only enzyme involved is RNA polymerase. It attaches itself to the promoter region, a DNA sequence indicating where it should start, and it builds from the 5 to 3 direction. So as you can see in this video, it builds from 5 to 3 using uracil instead of thymine. And just like DNA polymerase, it has a termination sequence, and once it reaches that, a hairpin loop structure forms that pinches away the forming mRNA sequence with the DNA and the RNA polymerase. So now we have our mRNA. So now that we have our mRNA, it must be fixed. And the first step of fixing is alternative splicing. Spliceosomes, as you can see in this video, splice out introns, uncoded regions of the mRNA. What we are left with are exons. Exons are the coded regions that exit the nucleus. The next step of fixing includes a GTP cap attaching to the 5' head. This helps it exit the nucleus and enter the ribosome better. The final step of fixing, we attach a poly A chain to the end of the mRNA. This chain is consisted of 100 to 200 adenine sequences, and this just helps the mRNA swim better. We receive a mature mRNA at the end, and now it is ready to leave the nucleus. The second step of protein synthesis, translation, happens in the ribosome. The mRNA strand moves from the nucleus to the ribosome. Translation begins with the assembly of the ribosome when the large and small subunits of the ribosome form to become one ribosome. The first codon, AUG, of every polypeptide chain attaches to the peptide site of the ribosome, and along comes a tRNA with the anticodon TAC. It brings along with it methionine, the first amino acid of every polypeptide chain, and in this sequence, the second is serine. rRNA, or ribosomal RNA, creates a peptide bond, as you can see here, between the methionine and the serine. The tRNA, as it reaches the exit site, leaves, and here comes another tRNA. This brings along with it the amino acid tryptophan, and again, rRNA creates a peptide bond between them. And again, as it reaches the exit site, it leaves. So this is called elongation. We are extending our polypeptide chain. But here, we reach a stop codon, signifying the end of translation. So yes, this is very short, but it was translation nonetheless. We have a polypeptide chain, methionine, serine, and tryptophan, and this is the end of protein synthesis.